Hello and welcome dear viewers to the another topic on drug degradation and impurity formation pathways. To develop a stable formulation, we have to understand how the drug degradation happens and what are the impurity formation pathways so that necessary measures can be taken to avoid the impurity formation or to avoid the drug degradation and the formulation can be made stable. So let's start with the video. See, stability of the drug and the drug product is of paramount importance in the pharmaceuticals formulations. Stability of the drug and the formulation is very critical considering the safety and efficacy. The drug should be safe and the drug should be having the required efficacy or the claimed efficacy. And for this, the product should meet the predefined specification throughout the shelf life or expiry period, and considering the drug substance, it should meet the specification in the retest period. So what is the shelf life or expiry period? The period in which the drug formulation shows or remains within the predetermined specifications and is safe to be used or consumed by the patient. Degradation leads to decrease in potency, that means loss in assay, increase in the impurity levels, loss of product design sometimes, and the product does not meet the quality specifications. Now see, loss of product design means if the product is formulated as DR formulation, that is delayed release formulation, or the formulation has specific release profile or dissolution profile, and if that is not maintained throughout the shelf life, then that will be called as loss of the product design. So these quality parameters are to be maintained throughout the shelf life. The quality specifications are must and must be shown by the formulation throughout its expiry period, because these quality specifications are required for the safety and efficacy requirements. Now coming to the quality specifications. So I have divided these specification into three types. First is physical specification, chemical specification, and the microbiological specification. So for example, physical specification is retaining the physical form or stability of the physical form. If you are making the tablets, that tablet should retain the tablet formulation form. The tablet should not get soft or hard. The tablet should not get discolored, or the capsule should not get discolored. Cream should not get melted. Aerosol should retain their particle size and the activation dose. Emulsions should not show phase separation. Suspension should be in the quality criteria of redispersibility. So these are the physical specifications. Then coming to the chemical specification. Now chemical specification means assay, impurity level, potency, dissolution requirements, and the drug release requirements. These are the chemical specifications. Also you can consider the water content or LOD and content uniformity. So these are the quality specifications which are coming into the category of chemical specifications. Then there is a microbiological specification. That means the formulation should be free from the microbial contamination, and this is very much required and very much stringent for sterile formulations. There should not be any growth of the microbes inside the formulation. Therapeutic and toxicological specifications. So out of these three specifications, if you consider the therapeutic and toxicological specifications, the drug performance or drug product performance like pulmonary formulations, aerosols, dissolution and drug release for modified release formulation and impurity levels. These will give you the therapeutic and toxicological specifications. Nowadays, nitrosamines impurities are attracting a more concern, and this nitrosamine comes into the therapeutic and toxicological specifications. So, out of these, for your information, you can remember the physical, chemical, and microbiological specifications, because based on these three specifications, the therapeutic and toxicological specification can be derived. Now, coming to the drug degradation and impurity formation pathways. How these impurities are formed, what are the mechanisms involved, and how the drug degradation happens. So, the drug degradation mechanism understanding is important to understand, so that this understanding and this knowledge will help to develop the stable formulations, to develop the therapeutically stable formulation. So, drug degradation and impurity pathways are physical degradation, chemical degradation, and microbial degradation. See, if there is physical degradation, the product may not be consumed or cannot be consumed. If there is chemical degradation, if there is microbiological degradation or microbial growth, then this product or formulation will not be acceptable for the clinical use by the patient. Now we will see the physical degradation. So physical degradation involves different degradation mechanisms like polymorphism, change in the solid state form or change in the solid form. Also, you can consider the pseudopolymorphism here. If there is change in the polymorph of the drug substance, then the biological activity will change, and this change is considered in the physical degradation. Then absorption and adsorption. This is mainly for the liquid formulations in which the container interaction may happen. Then particle size change. See, particle size change can happen into emulsion and suspension formulations mainly. Also, in some semi-solid formulation, this can happen. Then evaporation. If your product is stored at room temperature, and if there is loss of water from the formulations like aqueous formulations, for example eye drops, ear drops, injectables, and the liquid formulations packaged into semi-permeable containers, that time the evaporation of water from the formulation will lead to increase in assay, because the solution inside the container will become concentrated and it will have increase in the assay or potency. Then sometime evaporation of the flavors, and sometime the raw material may get evaporated. The example is evaporation of nitroglycerin. So this is regarding the physical degradation. Other physical degradation mechanisms are the hygroscopic nature of the formulation, deliquescent nature, or efflorescence nature. So hygroscopic nature means increase in the water content, and this increase in the water content may lead to degradation, change in dissolution, and the drug release. Then efflorescence nature and deliquescent nature will lead to loss or gain of the water from environment. And other physical degradation or physical changes involves the changes like hard cake formation in the suspension formulations, phase separation in emulsions, discoloration of the tablet or capsules, loss of flavor. Then loss of tablet hardness or increase in the tablet hardness, change in the capsule shell color, and leakage in the soft gelatin capsule. So here you remember that liquors in the soft gelatin capsule is a very challenging type of physical degradation or physical change in the soft gelatin type of formulation. Then coming to the chemical degradation. So impurities may get formed due to this chemical degradation. Chemical degradation pathways involve oxidation, photodegradation, hydrolysis, decarboxylation, and many more. So oxidation is one of the most important and obvious type of degradation. Oxidation involves auto-oxidation, oxidation due to the free radicals, oxidation due to the peroxides, and many more. Oxidation and auto-oxidation leads to impurity formation. Then what are the functional groups which are available or which are there in the APIs which leads to chemical degradation by oxidation? These are phenols, aldehydes, alcohols, and unsaturated fats and oils. Examples of oxidation are, or example of the chemicals or drugs which are prone to oxidation or sensitive to oxidation are ascorbic acid, certain vitamins, hormones, and antibiotics. Then comes to the degradation due to light, that is known as photodegradation, photolysis, or photodecomposition. In this, degradation is due to exposure of the formulation or drug substance to light, and this also involves many times the oxidation. Light sensitive APIs examples are riboflavin, furosemide, chlorthalidone, and many more. The next chemical degradation is hydrolysis. The hydrolysis reaction is there and it progress or it happens due to the presence of water, moisture, and is affected by the pH and temperature. Example here I have shown for the aspirin. Aspirin get hydrolyzed and get converted to salicylic acid and the acetic acid. So examples for the hydrolysis are aspirin with ester group, paracetamol with amide group, penicillin with lactam group. So hydrolysis happens and this leads to the formation of impurities and the degradation of the APIs. Then chemical degradation also involves isomeric changes. You might have known to the chiral impurities or racemic impurities or isomeric impurities. So isomeric changes or isomerism involves racemization, epimerization, geometric isomerism. So that basic part we will not go in detail. We will see how this isomerism has an impact onto the chemical degradation. So this isomerism may lead to increase in the impurity due to isomerism. These are known as chiral impurities. If your API has a specific isomeric activity and the another isomer is not having any activity or it is toxic, so that time this chemical degradation due to isomerism will have a much impact. The example is atenolol, warfarin, isopyramide, atropine. These are the racemic mixtures. And captopril, this is an antihypertensive API and it has two isomeric forms. Then another chemical degradation type is polymeric changes or polymerization. Sometimes it is also known as dimer formation or adduct formation. In this change, two or more molecules, that is known as monomer, may get combined together and these form the larger molecule, which is basically the impurity. Two API molecules come together and get attached to each other to give the impurity. Example of the polymerization is dimer impurities of apixaben. Example of polymerization is ampicillin and other beta lactam antibiotics. Then dehydration. Dehydration involves the loss of water molecule from the API molecule and which leads to degradation. If that API molecule is lost, that water molecule is lost from the API molecule, it will lead to degradation. Example is dehydration of the tetracycline antibiotics. Then another reaction is decarboxylation. So if the API contains this COOH group, that is carboxylic acid group, and if this carboxylic acid group is not available due to some chemical reaction, then this carboxylic group will be lost, and that loss will lead to degradation. Example is carbonicillin sodium. Chemical activities and incompatibilities with the excipient. See, generally the drug excipient compatibility is performed to have the understanding about the compatibility of the API and the excipients. These chemical activities and the incompatibilities of the API with the excipient or drug with the excipient may lead to impurity formation. The most known reaction is Maillard reaction. Reaction between the primary or secondary amine group of the drug and the reducing sugar like lactose causes color changes. This is known as Maillard-Browning reaction. Tetracycline also form complexes with the calcium. Drugs with acid functional group may get esterified with excipients containing alcohol group such as polyethylene glycols, known as PEGs. Then peroxide and the metal ions in the excipients can lead to oxidation of the drug. Some excipients like crosspoidones contain the peroxides. Some excipients like talc, magnesium stearate, lactose, dicalcium phosphate contains the metal ions, and these all together if present in the formulation may lead to formation of the impurities due to peroxide and metal ion catalyzed reactions, which mainly involve the oxidation reaction. So this is regarding the chemical degradation. Now coming to the microbial degradation. See, microbial contamination and degradation may happen due to contamination from raw materials, facility, equipments, and the personnel involved into the manufacturing. From anywhere this microbial degradation and microbial contamination can happen, and the quality management systems are there to restrict the contamination, but still some formulations are there which are very prone to the microbial degradation, like soft gelatin capsules are there, and some sterile formulations are there which are very sensitive for the microbial degradation. Microbial degradation may lead to loss of the potency also sometimes, loss or change into the physical form of the formulation, and increase in the microbial load or bioburden may lead to failure of the formulation in the stability studies. So for example, I have given here the microbial specification for solid oral formulations. So for oral solids, the total aerobic microbial count is tested, that should be not more than 10 raised to the power 3 colony forming units, that is also called as TAMC. Then total combined yeast and mold count, that is known as TYMC, that is not more than 10 raised to the power 2 colony forming units, and these are given into per gram. Then specified microorganisms should be absent, like absence of Escherichia coli. For some of the formulations, Staphylococcus aureus is also checked, Pseudomonas aeruginosa is also checked, and these are tested at the initial and at the shelf life period. So how to understand this CFU? Here it is given, this reference I have taken from the USP. 10 raised to power 1 CFU means maximum acceptable count is 20, and CFU is colony forming units. Second is 200, third one is 2000, and so forth. So here total aerobic microbial count 10 raised to power 3, this is 2000 colony forming units. Here it is 200, and the E. coli should be absent. So this is regarding the microbial degradation and contamination. So here we have seen the different type of degradation and impurity formation pathways. See, to develop a stable formulation, you should know how your formulation is getting degraded, how your API is getting degraded, what are the excipients, what is the manufacturing process, what is the packaging, and how we are going to make this formulation stable throughout its stability period or shelf life. That's why understanding the impurity formation pathways is very much important. So I hope this video might have given a good understanding about the impurity formation pathways. For the experienced people, this video will be like a refresh course, and for the less experienced or for the learners, it might have given a good understanding. See, the impurity related questions are generally asked into the interviews, and we should be in a readiness to learn the new things, so that at the time of interview or at the time of any examination, you will not run here and there. You will have a good understanding, and you will have a complete understanding and knowledge about the topics. See, some of the times in the interviews, the interviewer asks one question or two question, and based on that one and two question only, the interviewer can decide whether to select or not. So be prepared always for the interviews for your career growth and to excel in the professional life. Keep watching and learning the Pharma Learning in Depth channel videos. Share with your friends. Thank you for watching the video.